cell differentiation and stem cells.
In this presentation, we will discuss cell differentiation and stem cells in a simple and entertaining way. Firstly, we will discuss the concept of
Cellular differentiation.
what does it mean for a cell to differentiate?
Multicellular organisms all began as a single cell.
During the process of fertilization, called conception, the fertilized egg multiplies itself into 100 cells. This is called a blastocyst. Imagine the blastocyst as a company, and the DNA contained in the fertilized egg is the CEO. It tells all of the employees what their job is and how to do it. Each employee has a special job and must develop in a specific way to complete. So, this is differentiation. Differentiation. The process during which young, immature
Unspecialized.
cells take on individual characteristics and reach their mature
Specialized.
form and function. Blastocyst. The name for a fertilized egg cell after it starts to divide into more cells. Human blastocyst.
Differentiation can be affected by the environmental factors.
In the first month of conception, developing cells start to shift positions. For example, cells that are specializing in becoming cells of the fetus head move to one end of the embryo, while the cells that will become the feet move to the opposite end. However, when there are intoxicants such as alcohol present, the correct shifting of positions is compromised, causing potential deformities or damage. For example, the brain cells that were meant to move to the brain may not move correctly, causing brain damage.
Substances that affect differentiations.
Alcohol, mental retardation, physical damage, radiation, birth defects, childhood cancers, carbon monoxide, smoking cigars, asthma. Stem cells. What are they? How do they work? What is their function? As a blastocyst develops during conception, the cells begin to distinguish themselves into two groups. One group forms the placenta, the nutrient sac that protects the embryo. The other group forms the inner cell mass, which becomes the embryo. What does this have to do with the stem cells, you ask? Each undifferentiated cell, a cell that has not specialized its structure to serve a function, is called a stem cell. In addition, each stem cell is classified by what cells it is able to differentiate into. To put it in simple terms, each cell that is in the most basic forms has a limited potential to transform into another specialized cell, and the basic stem cells are classified into what they can become. Here are the different types of stem cells. Totipotent stem cells. This type of stem cell can develop into any cell in the human body or the placenta. Pluripotent stem cells. This type of stem cell forms the totipotent cells and can differentiate into any cell in the human body, except for the placenta.
Multipotent stem cells.
A type of cells that can only form into a closely related types of cells. Example, multipotent blood cell can only differentiate into other types of blood cells only, not brain cells or other types. Unipotent stem cells. They can only duplicate into their own type of cell.
Stem cells in medicine.
Can medical scientists use stem cells of one person to another? What benefits would that introduce? How? Is it ethical?
Cells follow a pathway of differentiation.
Think of a cell differentiation as a process. It isn't instant. Cells begin as their stem cells, then they partially differentiate. When a cell is not completely differentiated, it's called a progenitor cell. After the progenitor cell differentiates even more, it forms into its final form of a cell, present in the body.
How medical scientists are able to take advantage of progenitor cells.
From the previous slide, we know that the progenitor cells are incomplete in the differentiation aspect. What if specialists were able to use progenitor cells on a patient? That way, whatever a patient is in need for can develop and integrate with the patient's body and heal better. Wake Forest Institute for Regenerative Medicine has used cartilage progenitor cells and seeded them into a wax model of an ear. These cells will grow into an ear that can replace someone else's damaged ear.
Stem cells can be used to regenerate human tissue.
From the previous example of Wake Forest Institute's experimentation, we know now that medical scientists are able to grow new tissue with the stem cells to replace damaged cells. However, some aspects to experimentation with stem cells are ethically controversial. While healing a patient is prioritized, some people believe that life starts at conception, and using an embryonic stem cell is taking a life. Thank you for watching.